Hello! Let's look at the diversity around us today, and in particular, diversity in animal kingdom. Over here, you can see that there is an orangutan, a coral reef, and a sponge-like organism called as Aplysina fistularis. What is common among these organisms? These all are animals. How are these animals? They look very different from each other. In particular, we think that animals can move around, right? This can move around, but what about these? These are sessile, meaning they are attached to a substratum, hence they cannot move around. So how are these classified under the kingdom Animalia? Let's look into that. So an animal is an animal because it has a certain set of characteristics. For example, these are multicellular organisms, meaning that they have more than one cell. They are also eukaryotic, meaning that they have a nucleus and a bunch of double membrane bound organelles. Along with that, they also show motility. Now, this can move, but these two cannot. Well, they have larval stages since they show indirect development. These larval stages can move around and show locomotion, which is particularly important for their distribution around. So they show motility. Other than that, they have a certain embryological development pattern, meaning that they have blastopore, which either develops into an anus or mouth. If blastopore develops into mouth, these organisms are called as schistostomes. And if it develops into an anus first, these are called as deuterostomes. These two organisms are schistostomes, and this orangutan is a deuterostome. We humans are also deuterostomes. Let's look at the characteristic of animals that differentiate these organisms. So, what is the basis of classification under the kingdom Animalia? First, let's understand why classification is important. So, classification is important because we have a number of animals around us, number of species, of genre, of different taxons. So it is important for us to classify them because we can then understand that these two organisms are very distantly apart, whereas these two organisms are a bit closely related. Now this relation is important because if you want to understand the evolution of human, you are going to first look at an orangutan, then Aplysina fistularis. So classification is important because it helps us systematically study organisms. Now let's look at the basis of classification. Over here you can see levels of organization. Remember that all these organisms are multicellular. So how these multicellular or how these different number of several cells are arranged, that is the level of organization. There is a cellular level of organization which is present in Aplysina fistularis or sponges, where the cells are there but they don't really interact with each other. There is a tissue level of organization where these cells form tissues, meaning that they function in a very synchronized manner to get the job done. Then there is an organ level of organization where these tissues form organ, which is seen in case of Platyhelminthes. And then there is an organ system level of organization where different organs interact with each other to form organ systems, as in case of SK helminths, nematodes, we have annelids, mollusks, etc. So that is the level of organization. It helps you differentiate this from this. Then we have also this from this because they show different levels of organization. We have symmetry. Symmetry is when you cut an organism and find two equal halves. These organisms could be asymmetrical, as in case of sponges, meaning that if you cut this organism from any plane, you won't find two equal halves. It is also seen in case of gastropods that undergo torsion. We'll look into this later. Then we have bilateral symmetry, as in case of human beings, meaning that you can cut a human being in two equal halves from one plane. There is also a radial symmetry, which is you can cut an organism into n number of halves from any plane, and all of these halves will be equal. Then you have the number of germ layers. Now, all of these organisms are multicellular, meaning there are more than one cell. So, these cells are arranged in certain pattern called as germ layers. So, if there are two germ layers, an ectoderm and an endoderm, then that organism is diploblastic. And if there are three germ layers, meaning that there is an ectoderm towards the outside, mesoderm in the middle, and endoderm towards the inside, then that organism is triploblastic. Then we have the nature of coelom. Now, what is a coelom? Coelom is a body cavity between the body wall and the gut, where the organs are housed. So we have acoelomate organisms, for example, Platyhelminthes, where there is no coelom. There is a ectoderm, mesoderm, and an endoderm, but no coelom. Pseudocoelomates and coelomates. In case of coelomate, you can see two types of coelom pattern. We have a schizocoelom and an enterocoelom. So, schizocoelom is the coelom which is formed by the splitting of mesoderm. So, you have an ectoderm, mesoderm, body cavity, mesoderm, and an endoderm. In enterocoelom, this same pattern is repeated, but it is formed by the splitting of archintron, which is the primitive gut. In case of pseudocoelom, you can see that the mesodermal pouch are suspended. It is found in SK helminths. Over here you can see that we have segmentation. Now segmentation is metamerism or tegmatization. What is that? It is very much visible in case of an earthworm. You can see that there is an earthworm and it has a number of segments throughout its body. These segments are fixed. You can count these number of segments and they are species specific, meaning that there are a number of segments found in one species. For example, Ferretima posthuma will have, let's say, 50 segments. And all of the Ferretima posthuma will have only 50 segments. In these segments, organs are serially arranged and they are repetitive. Tegmatization is seen in arthropods, where there is a head region, a thorax, abdomen. This thorax has legs coming out of it and wings coming out of it in case of insects. So that is called as segmentization. Now, why is this segmentation important? This segmentation is important because, as in case of an earthworm, these segments form functional segments which are important for locomotion, forming a rigid hydrostatic skeleton. Also, these give a modular plan to body, meaning that if certain segment is lost, there are other segments which have serial arrangement of same organs showing redundancy. These are important and these are an aid to locomotion as well. Anyways, let's look at the notochord now. So notochord is found in chordates, but it is absent in non-chordates. It is a rod-like structure which supports body, helps in the locomotion, and in case of humans also, there is a vertebral column, right, which houses a spinal cord. Now, the chordate changes into vertebral column during the embryological development, so that forms vertebral column. So chordates are vertebrates or invertebrates, and there are non-chordates which don't have this vertebral column or this notochord at all. So that was about the basis of classification. How are you differentiating all these organisms from one another so that you can group them into certain organisms that are very much similar and certain organisms that are not. So let's look at the classification of kingdom Animalia into certain phylums. So phylums are, or the phyla are the groups that house organisms that are very similar to each other. So let's look at the porifera first. What are poriferans? Over here, you can see that Aplysina fistularis is a poriferan. It has a porous body. It has this big orifice, right? And small orifices throughout its body. So poriferans show a porous body. They are aquatic and mostly marine except Spongilla. Spongilla is fresh water. They have cellular level of body organization, meaning that their body is made up of cellular aggregates which don't form tissue. They are asymmetrical. Certain poriferans are also radially symmetrical, but mostly they show asymmetrical plan. Then they have a skeletal system which provides them with rigidity, and it is made up of spicules and spongin fibers. Spicules are hard things like this, and it is made up of calcium carbonate mostly. Then there are spongin fibers also, which are proteinaceous fibers. This could be made from silica also. And as you can already infer that spongin fibers are made up of mainly silica, because silica is the most stable mineral present on Earth's crust. These are hermaphrodites, meaning that sexes are not separate. Two sexes, mainly the male and female, they are found in one organism only. They also show internal fertilization and indirect development, which has a larval stage that can move around. And they have a very important feature called as canal system, which is also called as aquiferous system, and this is the sole system that sustains these organisms. So sponges have a perforated body, right? There is an inlet and outlet of water, but how is that possible? They are already present in water, but how is water entering and exiting? So this is caused by the beating of flagella of collar cells. So there are certain cells which have flagella that beat in a synchronized manner so that water can enter and exit. Now, why is it important? Why is canal system important? Because there is an inlet of oxygen as well as food, and there is an outlet of excreta and reproductive bodies, so that food and oxygen can enter along with gametes also, so that internal fertilization can happen, which can result in the formation of a larva. Over here you can see a collar cell which has a nucleus, a collar looking thing, and a flagella which beats so that water can enter and exit the body of poriferans. Over here you can look at the Escon type of canal system. This is the body of a sponge, and it is porous. It has this big pore. Over here you can see it has a big orifice over here. So same is shown in case of this. There is a big pore over here. Water enters through this pore called as ostium, and these pores are called as ostia. It enters into spongocoel, which is the internal cavity, and it exits through this, which is called as osculum. Over here you can see that the outer layer is called as pinacoderm, and an inner layer is called as coenoderm, which constitutes coenocytes or collar cells. Whereas in the middle you can see this portion. What is this? This is a mesenchyme. But this is a diploblastic organism, meaning that it has an ectoderm and an endoderm. So what is mesenchyme? Mesenchyme is not a mesoderm, but you can see that the mesoderm is starting to form. There is this layer which is very fluidy, very liquidy layer, and it has a bunch of cells suspended in it. For example, amoebocytes, which form all these cells. For example, coenocytes and cells present in pinacoderm. Over here, you can see another type of canal system called as simple cycon type of canal system. But it is different from this, right? Well, this is a very simple system. Here you can see that body is somewhat folded. This is ostia. It opens into a radial canal. So water enters through here into the radial canal, which is also lined by coenocytes. Then it enters the spongocoel and exits through osculum. So very simple. This is a bit complex than this. Then we have a diagram representing the same thing. This is the TS of a cycon showing cyconoid type of canal system. Here you can see that body is thrown into folds so that water can enter and there is more space for exchange of things, for oxygen, food, excreta, etc. Then also they show Sexual reproduction, meaning that gametes are developed. Over here you can see a gemule of Spongilla. Spongilla is a freshwater sponge. This is a very poorly stained slide, but you can see that it is circular in structure and it has these spicules coming out of it. Anyways, let's look at a few examples of poriferans. This is a Venus flower basket. It is usually gifted during marriages and weddings, and it is very beautiful. As you can see, it has a lace-like appearance, and this specimen is discolored because it was kept in formalin. And fun fact, formalin is carcinogenic, meaning it can cause cancer, but only when it is inhaled, not when it is touched or anything. Anyways, this is Hyalonema, which is called as glass rope sponge. And over here you can see that there is a rope-like thing and there is this organism. It is also porous and it has a cavity or osculum over here. Here you can see, well, this is SpongeBob SquarePants, which is Aplysina fistularis. So this organism over here, it is the SpongeBob SquarePants. Creator of this show, SpongeBob SquarePants, was a marine enthusiast. So he created this cartoon which was based on Aplysina fistularis. Anyways, that was about it for the phylum Porifera. You understood that these are marine organisms and mostly marine, and there are freshwater also. They show cellular level of body organization. Mostly they are asymmetrical. They have a skeletal system made up of spicules or spongin fibers. They are hermaphrodite. They show internal fertilization. This is the slide of a gemule. They also show indirect development because they have larvae and they have an aquiferous or canal system which looks something like this. And the TS of body wall of Cycon looks like this. Let's move on to Cnidaria. What is Cnidaria? Cnidaria are organisms. Cnidaria is a phylum. These organisms are mostly marine, just like in case of Porifera. They have radial symmetry and they are constituted by certain cells called as cnidoblasts. That's why they are called as cnidarians. What is a cnidoblast? Over here you can see a diagram showing a cell which has this. What is this? This is a cnidocell, which looks like a needle, right? So this needle-like thing is cnidocell. Let's say, and over here you can see that this organism, it is attached to a substratum. It cannot move around. So how is it going to defend itself from predators? What if organisms eat it? Right? So it will go extinct. So how is it protecting itself? As in case of plants, you can see that certain plants have thorns, and for example bougainvillea, and cucurbits family, they also taste bitter as they produce chemicals like caffeine. But what about animals? What can animals do? Over here you can see a cell, right? So this cell, it is basically the cell that protects this cnidarian from predator, from danger. What happens is, let's say there is a prey and cnidarian wants to eat this prey, but it cannot move. So how will it capture it? There is this presence of a cell called as a cnidocyte or a nematocyst, which is a stinging cell. It launches this cnidocell, a harpoon-like structure. This elongates and stings the prey, which paralyzes the prey so that it can eat it. So these cells are very important for their existence. Cnidocell, there is a spine-like structure also, very serrated, a stylet, and a operculum which encloses this apparatus into the cell. It also has a nucleus because it is a eukaryotic cell, and a mitochondrion for the persistent energy transfer. And this tube is usually coiled and it gets straightened out when it wants to prey. Then it has a tissue level of body organization. Poriferans had cellular level of body organization. So tissue level of organization means that the cells are arranged in a tissue which can function as a unit. These are also diploblastic, meaning that they have an ectoderm and an endoderm. Their digestion is extracellular as well as intracellular. Extracellular is through a body called a cell enteron. That's why these are all also called as coelenterata. And intracellular because over here you can see the body wall of a hydrozoan polyp. It has a cell intron, which is the median cavity, a gastrodermis, which lines the cell intron, and over here you can see nutritive muscular layer of gastrodermis. So this layer helps in the intracellular digestion. Anyways, they show metagenesis because they show polymorphism. Now what is that? Polymorphism is differences in the body form. Here you can see an obelia colony. It has this lateral extension and then this upright extension, and over here you can see a polyp which has hypostome, which is the mouth, and tentacles, usually in the number of 24. And what is this? This is a blastostyle in which medusa buds are formed, which then gets eventually budded off and form medusa. Now what is that? So obelia colony is a trimorphic colony or dimorphic colony usually, which has a polyp, a blastostyle through which medusa is formed. So one organism has all these different types of zooids which are very much different from each other, and they also function in many different ways. For example, the polyp is responsible for feeding. It is a feeding zooid, whereas blastostyle, it is a gonozooid, meaning that it is important for the formation of medusa, which is a sexual body. Over here you can see a slide of obelia colony. These are also called as sea fur because they are very furry and they are very small. It has a polyp, and blastostyle is not visible in this slide, but it has a polyp and these annulations are also present. So metagenesis is the alternation between polyp and medusa forms, which is the sexual medusa and asexual polyp form. That is possible because they show polymorphism. Over here you can see that medusa is formed, then medusa forms gametes which then come together and form this planula larva, which is free swimming. So that solves the mystery of how is it motile. Anyways, let's discuss a bit about how this medusa is formed. You will see that, and this is happening inside the blastostyle. First a protuberance is there, then a vesicle is formed, a bell rudiment is formed, because medusa is somewhat like this, a bell-shaped organism, whereas polyp is an upright bell like this, which is helpful in feeding. Then further development takes place, and finally a medusa is bud off. So that happens inside the blastostyle, and then through the gonopore medusa exits. Anyways, let's see a few examples. So remember I was talking about polymorphism. Physalia is an excellent example of polymorphism, which is also called as Portuguese men of war. Over here you can see a pneumatophore. It is not very much visible over here, but pneumatophore helps the organism keep afloat. There are gonozooids which are responsible for reproduction, a tentacle which bear nematocysts, these little organisms which are important for capturing prey and protection and also anchorage. Then we have dactylozooids, which are the skeletal systems which provides a rigid skeleton. So this is Physalia, which is an organism classified under the phylum Cnidaria. There is also this. What is this? This is a gorgonia or a sea fan. See, the polyps are shed off because this is also preserved in formalin, but you can see that this organism looks like a flan. And over here, what is this? This is a Gorgonia sea fan when it is living. Also note that this is colored. All of these things are colored. Anyways, let's look over here. You can see that there is a pen-like thing, a feathery pen-like thing. This is also called as a sea pen or Pennatula, which is its scientific name. It is very daunting to remember all these names, but if you keep reminding yourself of these names, then it becomes easier for you to remember all such names. Then what is this? This is a meandrina, which is a brain coral. Over here you can see suture-like things, and this is a dead colony. So this is called as a brain coral. Anyways, remember I was telling you about how sea corals can make their own food? How is that possible? Well, corals have a very interesting thing. They show inter-kingdom symbiosis. Symbiosis is the close association of two organisms, and this particular symbiosis is called as mutualism, where two organisms benefit from each other. And they show this organization, this relationship with zooxanthellae, which are algae. These are little small algae that live inside their cells that make food, which gives corals their beautiful, beautiful color. And it is also important for us to note that these can leave. This is a bleached coral. This is what a coral actually looks like without a zooxanthellae. And this bleaching happens because of global warming, increased temperature, and all the human impacts, pollution, etc. So this zooxanthellae leaves coral and corals become bleached. Now it is very hard for them to make food. Since corals are these rigid calcium carbonate things living on the calcium carbonate secreted structures, so it is hard for them to procure food so that they can sustain. But this zooxanthellae makes it possible because it makes food and these provide a safe haven for them. But due to pollution and all the anthropogenic factors that affect this environment, these zooxanthellae have started leaving these corals, leaving them bleached. But the good thing is these can again be reinvigorated so that they can allow the entry of zooxanthellae. Usually the coral bleaching is done because these expel out these zooxanthellae. Anyways, let's look over here. We have another phylum called as Ctenophora. Ctenophora is usually considered a minor phylum because the organisms under this phylum are very small in number. But let's see what it has. It includes sea walnuts and comb jellies. They are exclusively marine. They show radial symmetry just like this. Also, sea anemones, which are also corals, they show biradial symmetry sometimes. Now, what is biradial symmetry? Biradial symmetry is when you can cut the organism from two planes only so that you can find two equal halves. They show tissue level of body organization just like Cnidaria. They have 8 comb plates, and these comb plates are basically ciliary plates that helps the organism move around and perform functions. Digestion is also extracellular and intracellular just like this. They show external fertilization and they have indirect development because there is a larval stage. They also have colloblasts. Now what are colloblasts? So over here you can see that there is a cnidocyte or a nematocyst present. But over here there is a colloblast. Colloblasts are sticky cells. These are present in comb jellies and walnuts, which are secreted. These colloblasts are sticky cells that secrete a mucus layer that paralyzes, or basically it freezes the prey so that the organism can procure its food. They also show this property called as kleptonidae. Which other organism shows these property? Well, nudibranchs, we'll discuss that later. But kleptonidae is basically the property of ctenophores which is basically the tendency of these ctenophores to steal this nematocyst. Now this is not done done out of intention. This is basically done because kleptonidae is done because these ctenophores eat a prey, let's say, which is a cnidarian, and when these eat these cnidarians, their stinging batteries or nematocysts just pass down the digestive system and they are just sequestered there. These organisms, these ctenophores, don't use those stinging cells, but they are present in their body if they eat cnidarians. Over here you can see an example of cnidarian ctenophores called as Pleurobrachia. Over here you can see that there is a presence of tentacles and these comb plates, which helps this organism move around. Anyways, let's look at the phylum Platyhelminthes now. Platyhelminthes are also called as flatworms because they are dorso-ventrally flattened. There is no coelom present over here. They have bilateral symmetry and they are triploblastic, meaning they have an endoderm, mesoderm and ectoderm. They are acoelomate and they have organ level of body organization because they have organs like flame cells form an excretory system which is present for osmoregulation and excretion. Over here you can see a diagram of a flame cell of Fasciola hepatica. Here we have a pseudopodia and this is the cilia, which beat around so that excreta can enter and through excretory ductule it can leave the body. Over here you can see an example. This is the Fasciola hepatica, the anterior region of Fasciola hepatica, and this is a radii of Fasciola hepatica. So one thing about these platyhelminthes is that they are mostly parasitic. Turbularians or organisms like planaria are free living and they are very brilliantly colored. But let's say we have Fasciola hepatica or tapeworms and flukes. These are parasitic organisms which have number of larval stages. This in particular has a big tail which helps it swim and which is also important for its distribution. So this is Iridia. Over here you can see a Taenia solium. Taenia solium, as we have already discussed, is a tapeworm which is present inside the jejunum of an animal. So these platyhelminths show a number of larval stages. They are dorso-ventrally flattened, bilateral symmetry, triploblastic, acoelomate, organ level of body organizations, and have flame cells for osmoregulation and excretion. That was about it for the diversity in animal kingdom till phylum Platyhelminthes. We'll discuss other phylums in the next video. Thank you so much for watching. I hope I see you